Laura, so I'm interested, you were saying that you did start off in deep sea biology. What were you studying when you got a chance to take a dive on the Alvin, and what was your research looking at?
Yeah, sure. I was studying, there's a hydrothermal vent crab called Bithagrea thermodron. It's a little white crab that I think it's actually doesn't, almost blind crab that lives in and amongst the giant tube worms that are down there. And the crab is able to do really well in a high sulfide because these vents have a lot of sulfide coming out from underneath them, and they're able to live in a high sulfide and very low oxygen environment. And so my work was really trying to understand how they process that sulfide and turn it into a non-toxic chemical, and how they're able to live in low oxygen environments. So that was a really interesting project. I did my master's thesis focusing on that. And another project I had was working on a squid called Vampire Toothus infernalis, which is a really cool squid. I would love to see one. I don't know if any have been found on this expedition so far, but I know there are some on the Nautilus Live website. But I was also looking at the sort of the metabolism of those squid, because they're found in the oxygen minimum zone sometime, which is really low levels of oxygen, and sort of how does their metabolism work, sort of the ecological physiology to be able to live in that low oxygen environment. So it's been so long, I haven't thought back to my graduate school days for a long time, but fascinating at the
That's
time.
fascinating. Yeah, that's still fascinating. That's awesome.
I
And then
haven't
I
kept
also
up on the
heard if
research,
you,
but go ahead.
mm-hmm.
Oh, just I haven't kept up on the research. I'm sure so much more is known now on all of that. Lizzie, were you going to ask another question? I lost you for a second.
Oh, I was just gonna say, I feel like you might have told a story where you get initiated or christened almost when you do your first Alvin dive.
Mm.
Is that is that true?
You don't have to be. That's a
So it's, yeah,
volunteer thing. You don't have
it's
to
not
do that.
Yeah,
hazing.
do you want to?
I didn't use that word.
Well, this is
It's
what
like a
happened
rite
to
of
me. I don't.
passage.
Yeah. um, I I've heard of other things, and maybe you can elaborate, Bob, but I when I got out of the Alvin, they had like a bucket of ice cold water that was dumped on me, and I'm not sure what that was supposed to be, but you're down there for 8 hours, and you haven't had a chance to use the restroom at all that whole time, so the bucket of ice cold water is definitely a challenging thing to have thrown on
You
you
can
in
go to
that
the
moment.
bathroom.
Transit complete.
Yeah, actually, yeah, that's a
Thanks, Sam.
really interesting thing. You can go to the bathroom. They give you a bottle, and I think it's a
Oh,
little harder for females.
There's a funnel,
But I
an
remember,
adapter.
yeah, yeah, they give
oh
you
my
adapter. It's
gosh.
called I remember, a human element range extender, the
Yeah.
HERE bottle, and as as you're getting on board, they hand you this bottle, but you can imagine you're in there with three people, and it's this kind of tiny space, and you're laying down, and like there's no way. I actually knew what was coming, and my bladder's weak anyway, so I like rode the bike on the ship. There's a stationary bike the night before to kind of dehydrate myself so I could prepare for a day. Yeah, so I never used the
Wow.
bottle, but I'm sure you did after 120 dives.
Yeah, but I never had to do the other thing.
Oh my goodness, yeah. I don't even want to think about that.
Yeah, I've been in there when that happened, but I never
Wow.
drew that straw. Thankfully.
Yeah,
That's
you got to
funny.
hold off on your fiber.
All right, so we're not on
This-
transect anymore. So if anyone wants to zoom or look at anything, just let us know.
We'll just keep taking some 30-meter steps down to 5I, staying in this contour.
So I was in the submarine with Mandy Joy, and we were down in the Gulf of Mexico, and a storm kicked up while we were down, and the winds got up to like 45 knots. And they called down and said, "Hey, we got a little bit of a weather pattern coming through. Maybe you guys ought to just sit still for a while." And we sat on the bottom and turned everything off.
Oh my
And
gosh.
so
Wow.
Mandy she was adverse to using the HERE bottle.
How many hours were you down there?
Well, so it started to get dark, and they made the decision to come up. But before that, it was like they called down and said, "Well, you might have to spend the night." And her eyes got the size of dinner plates. Like
Oh.
there was no way she was going to
Oh
use
my
that bottle.
goodness.
But yeah, I don't think you'd make it overnight without using the bottle.
How long
way.
can the sub stay underwater if you do have to?
100 hours?
100 hours. Okay, so plenty of time.
Yeah.
Oh,
Not
wow.
that you'd want to be in there that long.
No.
Especially when you turn everything off, it starts to get pretty chilly in there.
Yeah. Yikes. And you were turning everything off to save
Save battery power.
battery power.
'Cause, you know, at that point, we didn't know how long we were going to be there. So we came up in the middle of that storm. It was pretty rough.
Yikes.
Yeah, we lost the science basket.
How many?
Science basket.
How many hours?
Well, we came
How
up.
many hours were you down there?
We came up before sunset. I mean, it was dusk, but yeah. So it wasn't that much longer than a regular dive, but it could have been. It was a very rough recovery, as you can imagine. The science basket broke off and was hanging by the safety lines. We actually didn't lose anything.
Oh, wow.
Yeah.
As pilot, were you able to stretch out, or were you stuck in that seat?
No, there's no real stretching out. Yeah, the pilot's sort of sitting on like a really low stool.
Right.
Yeah.
So if you're stuck there because of a storm for 24 hours or more, you're stuck on that stool?
Well, yeah, I mean, there's not really a lot of room to maneuver around in there. And you got to stay awake if you're the pilot. Well, got to keep the oxygen flowing. What's that?
You're the pilot. What is that?
What is that on the right? Yeah, the white.
We're going to investigate.
Is it a giant tunicate? Sure looks like it.
It's floating above something, but it looks like it's on a stalk.
Oh, wow.
It's a little spooky.
Chad and Erica from Monterey Bay are saying horsehead tunicate, stalked tunicate, culeolus.
It's huge.
Wow.
Wow. What a monster.
Somebody living in there.
Or
Several
being
somebody.
consumed in there.
Oof. I see why they call it horse
Wanna
head.
zoom
That's
in now?
fascinating.
Whoa.
So is that attached or is it just floating?
It's attached.
Yeah, you can see its base back there. Whoa.
That's pretty
Wow.
awesome.
Wow. There's a polychaete in the bottom. Looks to be commensal.
Yep. Looks like a scale worm maybe too. Very bottom.
Yep.
Oh, you mean that's the, yeah. What about the little tube there? You think that's part of the
It's part
body
of-
itself?
I don't know. I think that might be part of the anatomy, but
Wow.
I'm not sure. First I've ever seen one of these.
So what are they eating down there?
You got a really long, thin.
Yeah. Oh, little
Anchor
tube worm
point.
living on the back.
That debris looks really weird in the background too, what it's attached to.
Yeah.
Some kind of rock.
The lasers are on. Yep.
It's such a tiny little stalk that's holding it on there.
Yeah. I
But
wonder what
it's
that's
like
made of.
nylon cord though.
So how big is that? Oh, there's the lasers.
We had the lasers on when we came in. It was about 30 or 40 centimeters tall, or close to 30 centimeters tall.
20 wide there.
Yeah,
The wide point.
that's huge.
And that's interesting. Where would, like, are there intake openings? Like, is that the exhaust, or is that like the one and only opening to the outside? Because I'm wondering if this is the way the current is going. It's not directly in contact with the current, so I'm wondering how easy it is for it to catch and eat.
I'm trying to see the horse head
Oh, I
and
see
why
a
it was
horse
named
head.
horse head. You do? I
Yeah.
don't know. I'm tilting my
We're
head
looking
around
straight
trying to
at
see
it,
that.
like the opening being the eyeballs.
It's definitely one of the strangest things I've seen on the seafloor. It's
Doesn't it-
large too, maybe
See,
40
it's got like a
centimeters?
neck back there.
Yep. It's an incredibly long
I don't
stalk.
know. To me,
And
it looks exactly like Admiral Akbar.
yeah,
Yeah.
yeah.
It needs a new name.
Chad is saying it, the horse head is an unofficial name for it.
Hmm.
So maybe you can get the Akbar thing in there.
I mean, I see it though. It looks like a horse head with a short nose, and it has like, who's the guy from Star Trek? Is that Star Trek that has the band across his eye?
Geordie LaForge.
Yeah, a horse with one of those.
Okay,
It makes sense
gotta
if you
move
don't
on.
think about it.
All right.
Moving on.
Yep. Wow, that is enormous. One of the largest things we've seen down here.
Gonna be stepping off some ledges here, it looks like.
Huh. Interesting. Bridge nav.
Going down.
30 meters,
Neat.
bearing 160.
I feel like we've seen some of these ledges in previous watches. Like, this is pretty characteristic for high slope areas.
Looks like the terrain's starting to get more interesting now.
Definitely agree. It's in moments like these where I just, what's just to the left and what's just to the right, like what's just out of view that could be amazing and attached to this hard substrate that we're just don't have the field of view to see. Just to make sure we really are in the most interesting portion of this habitat. That's always what, there's a pull. And I just wish we could see it all. FOMO, I guess.
Fear of missing out on hard substrate.
Mm-hmm. Or like the giant sponge that's just out of view, but we'll never know.
Exactly.
We could change heading to like 170 and then be kind of looking at it a little bit more.
Oh, more like crab walking along
So is this the very
it?
first time that the Santa Lucia Bank is being explored?
I believe at these depths. And with this technology, if there might have been trawls or there, surveys that I'm not particularly aware of, but as far as like ROVs at this depth, I do believe that these are some of the first surveys of Santa Lucia Bank. And Meredith makes a good point. If we were to take a Niskin, like maybe we would be able to figure out if there were species in the area that we're not seeing, because the eDNA would tell us, hopefully. Unless it's an unknown giant sponge, to which Abby is replying, because then we wouldn't have the genetic bank to be able to identify. It would just be unknown DNA.
Are we collecting any samples for eDNA on this trip?
Yes.
This leg?
Let's see what we've collected so far.
So wait, does that mean that we
There's
want a
gold
Niskin?
urchin.
I don't think, unless Meredith says in this particular era, I think she was just giving an example, like we could address the FOMO by taking eDNA samples, but when I gave the example of like, well, what if there's a giant sponge just out of view and we'll never know, that would not be able to address an unknown sponge because we don't have the genetic bank with which to compare its potential eDNA to, so we would never know what it was, which is also unsatisfying.
Meredith is saying, sure, take a sample. Why not?
Yeah, especially in this rocky area, we might find that there are some hard corals here, the primnoids and black corals. And I don't believe we've taken many.
We have 3 available Niskins left.
Yeah, so we've only taken 3. Okay. Yeah, so whenever the ship is in position and it's safe to do so, Meredith said we can definitely take a Niskin here.
Oh, yep.
Target is going to be Niskin 4.
Roger. You don't need to stop, right? I was asking you.
You don't need to stop.
Okay.
Just keep going.
Yep. Perfect.
Wow, this is pretty darn steep.
Bridge nav. 30 meters, bearing 160.
So for those of you watching, we're about to take a water sample with a bottle known as a Niskin bottle. And essentially these bottles are open throughout the entire dive. And what the manipulator arm is about to do is pull a loop that's connected to the lids of these Niskins, which will trigger it to close, and therefore capturing the water that we're in right now and any environmental DNA that might be in it.
Which one are we going for?
4. The orange, 4.
Yeah.
So if you're looking at the quad view or channel 3, you'll see all the Niskins lined up with a few that have open lids. And then as the arm tugs on this loop, it's going to trip that lid to close and capture the water. So quad view is a great view for this sample collection if that's what you're interested in watching.
All right.
Thank you very much.
Zero 071, Steve.
Correct.
Copy that, 071.
Jump zoom out.
Lizzie, do you know how Santa Lucia Bank got its name?
You know, I don't. I wonder if Guy Cochran is still on, if he knows, or if anyone else in the science chat knows, but I am not sure. Oh, in the science chat, they're saying a giant cusk eel went by. I missed it.
Oh, I didn't see that one either.
In the science chat, just to give you an idea of who we're talking to and roughly how often, I check the science chat every few minutes just to see, as not someone who's super familiar with deep sea identification of various taxonomy, so fishes, corals, sponges, and invertebrates, in addition to listening to those on board like Steve, who know a lot about these invertebrates, and Sam, we have Tom Leidig with the Southwest Fisheries Science Center, who knows a whole lot about deep sea fish and corals and sponges. We have Erica Frew from the Northwest Fisheries Science Center, who also knows a lot about the same. And we have folks from the Monterey Bay National Marine Sanctuary who regularly, well, more regularly than us anyway, visit these depths off of their sanctuary, Davison Seamount, who are also really great with their deep sea identifications and have been helping us a lot with some of these organisms that I've never seen before. So Erica Burton and Chad King and Lisa Wolnick from the regional office for National Marine Sanctuaries. She's been doing a lot of learning in the short time that she's been a chief scientist with me. It's both of our first times, but she's gone in and looked up some organisms that she didn't see or wasn't able to identify on her shift, and she's coming in with IDs as well.
We're getting some responses from Lisa and Chad about the Santa Lucia name, saying that the Santa Lucia mountain range runs on land from Carmel to San Luis Obispo, so perhaps it's named after that. And most of the Big Sur coastal mountains are the Santa Lucia range, so.
That makes sense.
Yeah, totally. Thank you from the science chat room.
Bridge nav. Step 30 meters, 160.
And this area that we're in is just off the coast of Morro Bay and is nominated as a potential National Marine Sanctuary, the Chumash Heritage National Marine Sanctuary. And so we're learning about this area. If it does indeed become a National Marine Sanctuary, it would be, you know, having this habitat characterization and understanding the different species and what we're seeing down here would be, you know, help inform that. And this would, if the proposed sanctuary happens, it would be between the southern border of the Monterey Bay National Marine Sanctuary and sort of the northern area of the Channel Islands National Marine Sanctuary. So it would fill that gap in there. And Lisa's saying in the chat room that we've reached out to the Chumash and asked them for some names of the dives that we just did that would honor those Chumash connections to this special place. So stay tuned for that. We had Violet, who is her family actually nominated this area to be a National Marine Sanctuary, and she was on our Nautilus Next event yesterday, and it was just fantastic to get to know a little bit more about her and her connection to the ocean and sort of their reasoning and why they've nominated this area to be part of the National Marine Sanctuary System. And Violet Sage Walker is the vice chair of the Northern Chumash Tribal Council.
And that recording of that event is available on the Nautilus Live website under the education tab. Recorded both on Facebook and YouTube. Agreed, it was a great event to be able to hear more about this region and the cultural heritage that the Chumash are trying to protect and pushing forward the nomination for the sanctuary. So definitely.
Can we look at that coral that's sticking out at 3 o'clock?
Oh yeah, great spot.
Just want to confirm that it's what we've collected twice now and seen a few times.
Zoom in now.
Yeah, it looks like another Radicipes.
What's wrapped around it? Is that a brittle star?
Yeah, some colonies have been associated with brittle stars. The first couple we sampled, I don't think, are or have not had any associates on them. So this is actually a very interesting observation for coral associates with, or those invertebrates that form commensal relationships with this coral.
Here we got it.
All set.
Oh,
Did you get a laser shot on that? Or can we do a laser on the way out?
yeah, zoom out.
All right, good shot. Yeah, we've made a couple of collections of Redicipes so far. The deepest one was around 3,500 meters last night, just as we were ending our watch. At the end of a transect, we encountered one. And then just in the past few hours, between the last watch and this one, we've seen a couple of them.
Oh, that's another one on the ledge up to the right. Can barely see it now, but we'll pass over it.
Yep.
There's a curlicue one up here.
Oh yeah, third one. It's a good
And
example
then
of the small amount of hard substrate that some of these corals need. Sometimes just a small pebble or cobble-sized rock.
okay, so these are, because I know Tom was saying, and the difference between the two of them would be one row of polyps versus two, and then that would make them two different things, species. But maybe also if they're in soft or hard would also tell us if they were different.
I'm not sure about that for Radicipes. I think it's only a single row. I'm not familiar with any other species in the genus that have multiple polyps, or have what we would call whorls, in which case there would be multiple polyps paired.
Yeah, Tom was saying this morning they zoomed on a few, one that had 2 rows of polyps versus the one that we just saw had 1 row of polyps. So he wasn't sure if it could potentially be a different species or a variation in morphology. He's unsure.
Looks like that's a crab there in the distance.
Yeah.
Nice to see some hard substrate coming up.
Yeah, this is exciting.
Any chance to look over here at about 10 o'clock?
Yep.
I think this is similar to the sea fan, the primnoid fan we've been seeing lately.
This is an interesting variation in color here on the 2 different faces of rock.
Looks old. It's old rock.
Yeah.
Heavily altered.
Got a couple of different types of corals here. I'm seeing a clavularia-like coral, or maybe those are worm tubes over on the left-hand side also.
Yeah, zoom in out.
Oh yeah, clavularia. Oh, cool. Guy Cochran with USGS is letting us know that the outcrop is brown where there's a fresh break in the rock, but the black area is where it's weathered chemically. So it's a bit older since it broke off or fractured.
Interesting.
This looks like Narilla to me. Seems to be similar to what we've been seeing the past few minutes, few hours actually. There was a sample of this collected earlier. and it was tentatively ID'd as Calyptrophora, but looking back at the photos, it looks like more like Narella, but we'll be able to confirm for sure once we get it back on shore or on deck.
Hey, gotta get going again.
Yeah.
Time to go. I wonder if that's a holdfast back
So
there. Or is it
Meredith
just-
might be interested in knowing that the unbranched or very low branching coral we saw yesterday that was pretty common in the high slope environment towards the end of the dive was species of Narella. I haven't had a chance to key it out to species yet, but at least we know that it is Narella and either a unbranched or very sparsely branching colony from no species.
Can you zoom in now?
Was that expected it to be not so branched, or is that kind of like a new morphology you're not used to seeing?
Unbranched
Imagining things.
I think we're imagining.
primnoids are pretty rare. There are some species, but none that I think we could recall from the Eastern Pacific, Northeastern
like
Pacific offhand. So
Just a
it's
stick.
going to take some more digging. My supposition is that
Gotta go.
this is probably a branched Narella. It's just
Hey,
that
Colin.
they branch very late, or they branch very low on the colony. It's not always obvious if their certain morphologies are branched or not. But we only got a piece of the unbranched version, so I think we'll have to make some assumptions there.
Interesting.
The one thing that was pretty striking is that it had a very high density of polyps per whorl, about 12 polyps in a whorl, which is quite high. There's a species from the Atlantic that has a very high density polyp, high polyp density per whorl, Narella verslacei, but I don't think it's ever been reported from the Eastern Pacific or the Pacific at all. However, it's been observed in the Caribbean and the southeastern U.S. and also in Europe. So we'll
Zoom out.
be able to tell more once we get it back on shore, do some sequencing and some morphology, look at the sclerites a little bit more detail. I haven't set up the microscope yet for this cruise, so I don't know if we have the capacity to do any more higher resolution images, but would be something to look at once they get it back on the shore.
Is that better? No, I don't think so. In fact, it's been moved out some. So we didn't have the camera all the way out. That was part of the big problem there. You want to zoom back out again? Look around. Yeah. I mean, I'm centered up right there. Mongo looks like it's all the way outboard, so. Zoom on out. Is it better? No? You don't like it? Okay. I'm good with it. I'm okay. Use my sixth sense. What was the bearing?
160.
One.
Same we've been on for the last
It may be, but I don't remember.
hours. I need a telestrator. Just write numbers on it.
You need this to have a map and a goal, and then I would just drive to it.
Actually, the targets are in there.
Have to converse much at all. You
Oh,
just
perfect.
Put it up there
It's great for me too.
Just go. All right. Right. Just think of the bonus there.
Technically, the waypoints are in there.
They are, but like I
know.
cannot see that
I know.
to save
No,
my
I mean
life.
they're in here too, but we're just far enough away you can't see it.
Oh. What if you could draw a line? Huh?
That would
Can't.
be great.
I'm not saying you can. I'm saying you should.
See, look, targets.
You know, I actually wrote nav software for Alvin that did all that stuff.
So what you waiting for here?
Because it was a lot of work. I had a lot of time sitting up in the top lab. Yeah.
Is it possible to follow this higher slope? It looks like it kind of decreases in depth as we swing for south.
Sure can.
And then we'll hit this, what looks like the little valley of not steep slope at all. And then I'm thinking we should target that little orange spot of very high slope where the bathymetric line underneath it almost points to it.
Yeah.
Yeah, if we can hit that
Okay.
eventually. I'm not sure. I think we're going to have to pull not soon after the watch shift. So I don't know if we'll get there, but we can aim to get there if we have time.
Probably. Yeah, we'll change our heading to about due south.
Awesome. Thank you.
Yeah, actually, I think this will, you can put up here like a range and bearing to a target, you know? We just don't,
Bridge
we don't
nav.
use this as much as you can.
30 meters, 180.
And then if we don't see anything, we want to collect on this next 30-meter ship move. Maybe we can set up for a transect after that ship move is complete.
Copy that. Yeah, we've gone 200 meters, so no problem there.
Awesome. It's funny, as things come into view from the very top of the screen, the shadows make it hard to see what exactly it is and what the bottom is like, and I always think things are just floating.
What's this white?
You zoom in now?
Yeah, that's what I was thinking. It almost looked like it was standing up.
Starfish.
It's almost just lying over a rock.
Oh,
Okay, zoom out.
a little edge.
That's exactly what I was talking about.
Yep. Wow, that was
That's just a chubby coral? No, it's just a sea star.
quite a surprise.
Just a sea star. You can tell you guys have been
You
on
can
watch for a
tell.
long time.
Yeah. Yeah, I can't wait to see the highlight video that will hopefully include the coral garden that we saw. Oh gosh, was it 2 nights ago? When we were having a really hard time moving Herc up slope because the current was so strong. I think that was in the morning. Oh no, that was in the morning the other day.
I think that
kind of moves the stick on the meter in terms of what I'm impressed by. I'm starting to sound like Steve.
was that yesterday morning, I think?
Was it yesterday?
Not sure.
It's like days ago.
Had a couple of watches since then.
It's like you're here on the ship, isn't it?
Yep.
Except without the delicious buffet.
When you said we were in the Channel Islands last week, I was like, are we sure about that? I'm pretty sure it was last month.
Yeah.
Yeah. Yeah.
I thought you were
So
going to
you
say you were
folks out
excited
on
to
the
see the highlight video from this dive. I was like, really? You
That brown
can't
sea
wait to relive
cucumber.
it? Yeah.
All those highlights of the 3s and the 4s.
So for Lizzie and I are talking to you from our living rooms here, but for those who are out on the ship, how long have you been out there?
Who's
For this
been out the
expedition,
longest?
days or weeks?
Well,
Robert?
I wasn't on the ONC leg, but I was on the shakedowns, and I'm doing all the rest of the legs, so all but one.
So since you were out in March, got off for a little bit, and then, oh, not March. That's when we were supposed to have shakedown. August?
Yeah.
It's hard to remember the months.
I think it was August, right?
Yeah, it was.
And then we got back to the ship September 19th.
Yeah, well, I did 2 weeks in quarantine. I've been home for 6 days for that.
Yeah.
Yeah.
And then a couple of us came on the 2nd of October. Steve, how long have you been out?
It's been 87 years since I stepped foot on land. No, I got out on the 19th of September.
Okay.
Yep.
And that
After
was
2 weeks in quarantine,
Yeah.
so I've been away from home since just after Labor Day.
September
And that's
4th.
Al too?
Yeah.
I go
Quarantine
home in
buddies.
December.
Yeah.
3 months.
And I was planning
most
on
of you
having
are-
a light year.
Are most of you getting off in San Pedro after NA123?
Yeah, we've got 11 people off signing and 14 on signing for the final ROV cruise of the year.
14 on signing?
14 on signing, and some of those folks are staying for the month of mapping that's happening that runs until December. So, but there's
Got it.
many scientists and two lead scientists and several grad students who are coming on from Scripps for that final ROV cruise. And then we're having a reduced hour ops, so 8-hour ops, in order to process a high level of samples. So slightly different pace.
So only 4 ROV people. In that
Yeah.
leg.
Yeah. Duplicates of every role instead of triplicates. Bridge nav. 30 meters, bearing 180.
So are you saying that there's going to be more ROV work in December?
No ROV work, but just mapping, a month of mapping to wrap up the season.
Oh, I understand. Got it.
And Sam, you're multi-talented. You do video and navigation and science communication, and what other things do you do?
She changes the gripper on the manipulator.
Did that. Got some crane training in. Yeah, this year's looked a little different. I'm normally the communications manager, so managing our social media website, which we just overhauled over the last year and launched in April. All of our press outreach, and then our managing our video production team on shore. So it's been, oh, and then coming out, usually I'm out as a communications lead on the ship, but since we were reducing staffing this year and made the very difficult decision not to bring out science communication fellows, we also did not staff for communications leads, so ended up coming out in the navigation role, and it's been really great to be able to kind of bridge all the different kind of situational awareness, as I've been saying, of all the other roles, and being able to use that, instead of using that to communicate externally with the public, using that to make sure that science ops and back deck ops and bridge are all on the same page. But yeah, I have, it's kind of an ongoing joke now that I think I've sat every roll in the van at least for a few hours, if not for a couple weeks, except for Herc. But I did drive the arm the other day.
Nice.
That's really cool.
Actually, you could do this from your seat.
Yeah?
Yeah.
Well.
Just got to push the buttons.
That's all it takes? You just got to push the buttons?
Right now it does.
Well,
Yeah.
add that to the list.
So I guess it is a robot. It's in robot mode right now, so it could be called a robot.
Oh. Oh. So.
I heard a R word something.
Yeah.
Robert.
Robert.
It's a Robert right now.
Yeah. So it's in fully autopilot mode. But you don't call a plane a robot, right?
You could. I mean, what's the definition of a robot?
No, you can't.
It has to have autonomous. So a washing machine is a robot. You know, if you look at the definition of robot, yeah.
Yeah.
Because it, you know, it changes cycles on its own and, yeah, completes the tasks without you intervening.
Paul, if you call an airplane a robot, the pilot will get mad.
Well, same here. When you call the ROV a robot, the pilot does get mad.
Yeah. So it's as robot-y as an airplane.
Yeah, so we're back in the center now, which matches what we're seeing on the slope map.
Yeah, it's pretty accurate today.
Not very steep, not a whole lot going on.
Bridge nav. .30 meters, 180. Moving due south.
180. All right.
Time to push some buttons.
Actually, it looks a little bit warmer as you actually move up into the next contour, shore side. I don't know if you want to stay in that or you want to go back into the soft.
I think if I had heard Lizzie correctly, I think the thought was to move to the southern contour. That brings us closer to this orange spot that now has a target on it.
Yeah, that little hot spot.
Okay.
I think we're going to sacrifice slightly steeper slope in hopes that this one spot will hold. Wonderful miracle of corals.
That's going to take us a little bit down slope. That shouldn't be a problem, right?
Depends on how steep it gets.
I don't think it's going to be too steep.
Yeah.
Yeah.
If we want to move kind of down slope and then crab walk towards it, we could do that too. Or if we move a little bit southwest just to get to that depth contour and then follow it up. However you guys feel is best to aim for that target.
I think due south is fine.
Okay.
Yeah.
Yeah, nothing crazy has shown up, even, I'm assuming even on the radar or sonar, if you even have that on. Doesn't seem too complex, even in the steepest areas.
Yeah.
Nope. Yeah, looking pretty flat.
Yeah, and then
Light
if I'll leave it up to the watch after us. Well, I guess we still have an hour and a half. If we want to continue maybe slightly going down slope to hit the next orange hotspot, and there's kind of a cluster where if we can reach, yeah, that hotspot first, and then I'll head slightly down slope again to if there's like a It almost looks like a wall. It's where the contour line below that one had like points towards it. There's like a bright blue spot, and then right on top of it is a red spot. It almost looks like a little ridge.
I'm gonna drop a
There's just
target
like a
where I think
cluster of
you're talking
spots.
about.
Yeah, I think that's it. And they can change their minds if they want, but I'm gonna guess that might be the most next interesting target if they make it. Oh, what's happening?
Sounds like a robot.
Yeah, our government machines beep when they log themselves out for some reason. It's like, hey, get back to work.
I've been watching the oxygen saturation as we're cruising along here. It's at 20%. Is that pretty consistent at this depth, or does it vary much? I don't know who would be the best person to answer that question.
We're all looking at Steve.
Can you repeat the question?
Yeah, I've just been watching the oxygen saturation, and it's at 20% at this depth. Has that been pretty consistent, or?
Yeah,
do you want to?
I've been looking at the oxygen concentration since the start of our watch last night, just after we got on bottom. It's been about 100 micromolar since we got on bottom, and it's risen by about just a micromolar or two. Saturation, yeah, between 19 and up to 25%. It's consistent for this depth. It's more well oxygenated than the minimum zone waters above us. But you know, even when we were diving at Davidson, it was about 100 micromoles at a similar depth as well.
Can you can see a plot of that in Grafana, right?
Yep. Yeah, I can pull it back up for whatever interval we're curious.
Yeah.
So oxygen minimum was, let me see here.
Was that available on shore? I don't know how the new setup works.
Yeah, I think the Grafana is available on shore, but I'll have to, I haven't been on shore enough to use it.
Is this a helpful plot on Grafana?
Is that it?
That's the data,
So,
but it's squished into a flat line.
Oh.
Looking here at the oxygen profile for our descent.
Yeah, I can see that now.
And yeah, we bottomed out at about 2 micromolar, about 866 meters, and was rising again through 2,500 meters, and then at about 3,700 meters it flatlined. And it's been about between 110 micromolar and 105 micromolar since then. Percent saturation, yeah, so since we've been on bottom, percent saturation's been between 26 and 23%.
How does that compare to the surface? If someone
It's about half. So the surface water is about 200 to 300 micromolar. Yeah, so the surface data I have for 50 meters is 297 micromolar.